So hopefully you saw this preview lesson before I got into the two forms of testing that scientists can use. Because you're going to need this information right here, the previous video, in order for you to understand what I'm going to talk about, or at least understand a little bit better. So RT-PCR is, well, I don't need to type it, I already typed it out. Reverse transcriptase polymerase chain reaction. And there's a bunch of words, but you don't really actually need to know or memorize it. We already talked about what those letters mean, but basically reverse transcriptase is an enzyme. And polymerase, this part, bold that part, is another enzyme. What does this first enzyme do? It converts RNA into DNA. So if we saw that previous picture, it's this little enzyme, this little protein that moves around, attaches onto an RNA, and it makes a copy, but converts it into DNA. That's its main function. And then we have a second part. Polymerase is another enzyme. And what does that do? It makes copies of DNA. So why do we want to even bother doing this? Well, the problem is that when we collect a sample from someone's nose or someone's throat to try to see if they have coronavirus, do they have coronavirus? Well, the problem is that sample it's too small. It doesn't have enough of the not enough genetic material to tell. And the only way to get actually enough, if you're going to take directly from the source, is if you actually just put a human inside a blender, blended it up, and then took out those parts. But then you would have a bunch of other useless things mixed in, and you wouldn't even be able to tell if you got the right stuff. We choose the nose and the throat because there's a high concentration of that specific type of virus. So we take the sample, there's not enough. This is a process to make more of it. So we use reverse transcriptase, RT, to change the RNA into DNA because we can't really make copies of RNA without it breaking down. Then we have PCR. What is PCR? Well, it's basically this big machine. And I'll talk to you about how this big machine works. This big machine has these little slots. It's this metal plate with little slots where you can put little tubes of liquid inside. And so before, a long time ago, 30 years ago, when scientists wanted to make copies of DNA, well, they used it the natural way, where in our bodies, when we grow bigger, our cells split and make more copies of DNA. And bacteria, also another living thing, can do the same thing, but a lot more frequently. I think E. coli can replicate every 20 minutes. That means if you started with 1, in 20 minutes you would have 2, in 40 minutes you would have 4, and in 7 hours you would have something close to 1 million. So it's pretty fast, but still kind of slow because we need probably close to a little over like 500 million DNA strands for us to actually physically see markers that we attach onto it. So how do we get that much? Well, back then we just had to grow it for weeks. But today, deep in our oceans, not today, this is 20 something years ago, deep in our oceans, there's these organisms that are extremely resistant to heat. We stole their enzymes, and reverse engineered these heat resistant enzymes so we can, in our little machines, in our little PCR machines, we just do little drops of their enzyme in water to do what these bacteria are able to do, but way faster. We stole these enzymes. We need these heat-resistant enzymes because these machines heat it up to break these DNA strands in half, have these enzymes attach, and then cool it down. They make copies, heat it up, detach, make copies. So it's just a cycling from hot to cold, thousands of times. So it just keeps doubling over and over and over again without the enzyme breaking down. Because if it's too hot, then normal enzymes break down. So what does this get? This gets us billions of duplicates. And there's a few ways to see it once there's that much. It's still in a little jar. It didn't explode out of the jar because it's still very small. But there's two ways to see it. You could use a gel in which, because we know the features of DNA, it has a slight charge. You could send a current through it, and then bigger pieces move slower, smaller pieces move further. Not super important for right now, but basically there's things that we can attach to it, like fluorescent probes, glowy protein bits, and those are going to light up, and then we can compare what we see to the data that scientists have gathered elsewhere, in Wuhan, China, who did the initial research and found the genetic data for it. And we can compare, hey, does it match? And if it matches, it means, uh-oh, the person that we got the swab from is definitely positive and has that disease. If it doesn't match, that means whatever DNA, whatever RNA that we found most likely means that they actually aren't positive, they don't have it. Obviously, errors can occur. So errors can occur if there's not enough material. If we just got no viruses for some reason, we just mixed. We accidentally swabbed their cheek by accident. The outside of their cheek is not going to be useful. Or the test runner messed it up. Those are going to result in false negatives. I'm just checking the time on the video. So how accurate is this test? I'd say if you're doing it in terms of pure research and a scientist is conducting it, you're 98 to 99% accurate. And if you're not accurate, it's always going to trend towards the negative, where it says, hey, you didn't have this disease when you actually had it. So that is slightly dangerous. The second test that we have, that bigger, this one's a lot quicker. It's probably about 15 minutes because it relies on something called capillary action. So first, refer to the viruses and vaccine video if you see words that are confusing. I'm going to try not to use anything that's too complicated. So basically, you get a sheet of paper. Scientist grabs a sheet of paper. With the specific lab created or broken, let's be accurate with that. Broken protein pieces of the virus. Let's look at that picture again. So in our lab, let's see, we just chopped off this chunk, and then we chopped up a bunch of these chunks. We basically smashed up all these viruses and smeared it on a piece of paper. And they're dead. They're not capable of performing their actions of infecting people anymore. So you can't get sick from these tests. It's literally not possible. But the main focus is we want these little nodules on the outside. Remember what the word was? Antigens. So we smeared on that piece of paper. And that piece of paper is going to look something like, let's see, file, new. No, we do not want to save. It's going to look something like, wow. And there's going to be little bars. And I'll name it something like this. This is C, this is T, T1, T2, and then this is a little slot for you to drop your blood, which we'll use red for. So there's your drop of blood. You got your finger pricked, you drop the dollop of blood on here. And then you add some water. You add some water. It's actually buffer solution, so it's not contaminated with other things, but it essentially acts like water. So once you add water, there's this action known as capillary action. So because this is a piece of paper, it's going to spread through it. And what capillary action is, I'll get into that way more in a different video, is because essentially water actually has a charge as well. So when it touches certain materials, it's actually going to move along those materials if it's a certain shape. So this whole thing moves up, spreads through there. That's water, by the way. That's our goal. So what each of these stand for is, let's change colors again. As our blood moves into here, we put, or scientists put, some of those virus stuff over here. So when these two things mix, it's actually just going to change colors, something wild. We'll do purple. Oh, I changed colors. So if it changed colors, that means, uh-oh, you're probably positive. Why are there two tests? Because there's actually two different types of pieces that you could have taken from that virus. So it's just to make sure that this test is actually super accurate. And these two tests actually give a little bit more information, which I'm not getting into now about how it does it exactly, but it's able to actually tell whether you had the disease before or you currently have it based on which one of these two light up if some of them are missing. Again, not super important for right now. But yeah, they're just going to change colors because the two pieces, the blood and with your antibodies and the virus chunks that are smeared on there, the antigens, are mixing and binding, and we've attached some probes so the colors change. If these both light up, very, very likely that you're positive for coronavirus. What's the C for? C is for control. And we learned about controls in science already. Controls is something to compare it to make sure that we're not just testing for random things, right? So why do we need a control here? Well, imagine that we had another test. I'm drawing a really quick one. We had another test, and the water that we poured in and our blood, which is right there, only reached up to that second point. What's going to happen? Actually, let me take an eraser. Do I have an eraser? We're just going to have to go backwards. Okay. Separating, da, da, da. Let's say we dropped our blood. That's not red at all. Dropped our blood. Started adding some water, right? But the water only reached up to here. And our virus particles are all the way here. They're smeared over there, they're smeared over there, and then we have our control. What happens here is you're going to get a what? Pause the video, guess. Or is it going to be a false negative, which means that it says you don't have the disease, but you actually do? Or is it going to say that you have a false positive, which means you don't have the disease, but the test says that you do have it? All right, unpause when you're ready. So what's going to happen here is if it doesn't actually reach here, these aren't going to change colors. There's no color change. So you're going to get a negative result. This is a positive result. If you get a negative result and the water just didn't travel enough because you didn't add enough water, that's a huge problem. So how can we tell? Well, because we have our control, that one just lights up with a different color. I'll do purple again. With a different color whenever water touches it. So if this doesn't light up, it means you have an invalid test. Because the control didn't light up, it means the water didn't even reach it. Invalid. So that's pretty much how the second test works. It's a lot cheaper to make. All you need is really, some scientists, some paper, and some viral particles and some, tools in order to actually smash up the bar. Because you can't just smash it up with a hammer. You need to use certain chemicals and things to break them apart. And you can't use those chemicals to cure us because those chemicals would kill us too. But they can be used in the lab to break up viruses. So what errors could we make? We kind of already talked about those. If the water doesn't reach the top, it gives us a false negative. We could have had it or we don't, but it's just going to say negative automatically. And and if it's made poorly, which is something that I'm hearing is happening in some places, is that unfortunately, if they don't put the correct viral pieces, you could have something that's shared with another virus put in there. So if you had, hey, the regular flu, then it could be, labeled as positive. But it's impossible for any properly made test to show a false negative. So I'm not sure on the quality of some of these tests, but I do know that a quality made test should be close to, again, close to 100% accurate, uh, hovering around 98%. And that's it.